Question number 10 is a question that involve the critical thinking and also some concept of the FOV and magnification. One of the special thing in this question number 10 is that it is using the millimeter ruler. That means it's using the normal ruler instead of the stage micrometer in order to measure the FOV diameter. So it is something special. So, yep, sometimes you have to take note on this. Yeah, if let's say the questions is not using the stage micrometer, okay, they can use actually other thing to measure the the diameter of the FOV as well. This question is asking for the number of cell across the diameter. Okay, so meaning that we need to find the the actual size, actual diameter of the cell, and also the diameter of the field of view. So there are two parts. Yeah, so this part is looking for the actual size of the cell. Then only then we can know. Yeah, and only then we can know how many cells can fit into the diameter of the FOV. So for the first part, to solve the actual size, to find out the actual size of the cell, we can just follow the three same steps, yeah, in order to find the actual size, since the magnification has been given, okay, and also the diameter has been given. So based on this info, we can conclude that these questions is actually related to the estimation of the cell size based on the diameter. So to solve this, yeah, we will still use the same three steps. So step number one is we have to find the one pitch on the stage micrometer. Okay, so for this step, seems like because the questions is special, yeah, so seems like it is not needed. Yeah, it's not needed. Yeah, why? Because there's no stage micrometer being used. So that's why we cannot find one pitch on the stage micrometer. Okay, so this one, no need. Yeah, it's not needed. Okay, why? Because the questions is using ruler. And if let's say it's a millimeter ruler, so that means that this one is one division means that it is 1 mm. Okay, it's very straightforward, yeah, because it's using millimeter ruler. So this one line is 1 mm. Okay, so that is for step one, yeah. So we no need to do anything for step one, so we can skip the step one. Then we move on to step number two. So step number two is we want to find the diameter. Find the diameter of the FOV. So based on this figure, yeah, based on this figure, we can see that this is one line, two line, three line, four line. Okay, so that means that this is millimeter ruler. So this is 1 mm, 2 mm, 3 mm, 4 mm, because each of these is 1 mm, is a ruler. Okay, it's a line of ruler, yeah. So that's why its total is 4 mm. Okay, so that means this is the diameter of the FOV, yeah, the diameter of the FOV. Okay, so it is 4 mm, yeah, 4 millimeter. Okay, so same thing, we don't have to do any calculation, we just have to gather the information. But please be reminded that this FOV diameter, the 4 mm, yeah, the 4 mm FOV diameter is under 40 times magnification. Okay, it is a ruler view under microscope with 40 times magnification. Okay, so that means that this is under 40x magnification, 4 mm. Okay, why we need to state this? Because the FOV diameter will change. The FOV diameter will change. Yeah? The actual size, the actual diameter of the FOV will change according to the magnification. Yeah, so this one we will discuss further later in the concept part. So as for now, we proceed to the step number 3 first, which is to find the actual size of the cell first. Okay, so that would be step number 3. We continue with that first. So step number 3, we will find the actual size. Find actual size of the cell. So based on this, we can see here, there are 200 cells here that can fit end to end in this FOV along the diameter. So meaning that across the diameter here, you can fit in 200 cells. Okay, and we know that the diameter at this 40 times magnification is actually 4 mm. So meaning that this 200 cell is equals to the length of 4 mm. Yeah. So with that, we can write it down. So from the questions, we know that 200 cell equals to 4 mm. So same thing again, we will convert the millimeter to become the micrometer. So to convert to micrometer, we will times 1000. So it becomes 4000 micrometer. So we will do the conversion here. So that would mean that 200 cells will be equals to 4000 micrometer. And one cell will be having the size of 4000 micrometer divided by 200. 200. That is the number of cells. Okay, so that means that the size of one cell is equals to 20 micrometer. So now we know, okay, one cell, the actual size is 20 micrometer. This is actually the first part. Yeah, this is for the first part. So now we know one cell, the actual size is 20 micrometer, but we need to estimate the number of the cell that can fit into the diameter of the FOV when the magnification change. From 40 times magnification, now magnification has changed to 100 times objective lens. So that means that it will be times 1000 magnification. When magnification change, the diameter of the FOV will change as well. So that's why we will need the second part. Second part of the questions is that we have to find the diameter of FOV under different magnification, under higher magnification. So before we proceed to this part, this part B, we will go through the concept. The concept of this FOV actual diameter versus the magnification. So in the theory, what you need to know is that the image size of the FOV is always the same, regardless of the magnification. What I mean is that this is the FOV. So that means that what you see under the microscope, yeah? So this is the round shape that you see under the microscope. No matter what magnification you use, whether it is 40 times or 400 times or 1000 times magnification, you will always see the round shape with the same size because that is the image size, right, of the FOV. So under whatever types of magnification, the FOV is always the same size. But is that means that their actual size is the same? No, the actual size will be different. Image size is always the same, but actual size is always different. Okay, so what you can know is that, I will write it down here. When we zoom in, when we zoom in. So when you zoom in means that we increase the magnification. When you zoom in, you increase the magnification. We are going to see smaller area. So that means what? That means that the FOV actual diameter, the actual diameter of this FOV, when you zoom in, will become shorter. Okay, so that means that you see smaller area. So that means that the FOV diameter, the actual diameter, will be shorter. So on the other hand, when we zoom out, when we zoom out means that we decrease the magnification. So when we decrease the magnification, then we will see larger area. when you see larger area, that means that although the image size of the FOV is the same, what we see under the microscope, the round shape is still the same size, but then the actual diameter actually increase already. Okay, so remember this concept, and we can look at the diagram here, okay, to understand the concept better. So let me show you the diagram here. So this is the diagram. Okay, so we can imagine that this is the FOV. Maybe I. Okay, so let's say this is the FOV. Okay, so I will copy and paste here. So just now we mentioned that the image size of the FOV is always the same regardless of the magnification. So yeah, under the microscope, when you are using 20 times objective lens, this is what you will see, the round shape. When you are using 50x objective lens, it is still the same round shape. When you are using 100 times objective lens, it is still the same round shape. So that means the image size of the FOV is always the same. Okay, so now at lower magnification, yeah, at lower magnification, the actual size of the diameter is actually larger because you can see it's around like 0.6 mm. That is the actual diameter. But the image is still the same, right? Image is still the same. It's just the actual size is 0.6 mm. When we zoom in, that means that we use higher objective lens, okay, higher magnification. You can see that the image size is still the same, but the actual size is now only like 0.25 mm. The actual diameter has decreased. This one higher, this one lower. And when we further zoom in, we further increase the magnification. Now, the actual diameter of this FOV is only 0.12 mm. So same thing goes to the cell. Let's say the cell is 0.1 mm in size. Yeah, let's say this is a cell. When we zoom in, then the size of cell will become like this. It's still 0.1 mm. The actual size is still the same. Yeah, but that also means that the diameter of the FOV have actually decreased. So that's why the size of cell look bigger, but actually the actual size of the cell is still the same. So while under 100 times magnification, the size of cell is like this. It look huge, okay? But that also indicate that the FOV of the FOV diameter is actually decreasing when we increase the magnification. So based on this concept, we can conclude that the diameter, the image of the diameter of FOV is always the same. Image is always the same regardless of the magnification. What is changed is that the actual size of this FOV, you can see it is actually decreasing from 0.6 to 0.25 to 0.12. So this is the conclusion that we can make here from this concept. So once you understand this concept, then now we will proceed to to solve the part B, the second part of these questions. So from the questions, it tells us that now the objective lens has been switched to 100 times lens. Yeah, 100 times lens. 100 times lens means that the magnification is now 100 times. Then you have to times with the 10 times magnification from the ocular lens. Yeah. So with that, total magnification will be now 1000 times magnification. Okay. So based on the concept that we know just now, okay, so I will put it as step number 4. Yeah. So this is the extra step. Yeah, this extra step is mainly because this question is special. Yeah, it's involved the critical thinking. Okay, so now we need to find, we need to find the FOV diameter. We need to find the FOV diameter, okay, at 1000 times magnification first. This is what the question wants, the second part of the question. We need to find the diameter of this FOV at 1000 times magnification. So from the concept, okay, maybe I write it down. So we know that the FOV that we see, right, is always the same size. Yeah. So the image, that means that the image size, the image diameter of FOV is always the same size. Okay, it's always the same size regardless of the magnification. Okay, so based on this, what we can do here is we can apply the formula, because when it comes to magnification, it's always the image over the actual, right? So based on this, we can know that image is always equals to the magnification times the actual size. Okay, so this is what we have. So since from the concept, we know that the image diameter of the FOV is always the same. So that means that image is always the same. Yeah. So no matter the magnification is 10x, it's the same. Or if the magnification is 100 times, it's still the same. Or if the magnification is 1000 times, the image size is always the same. So based on this, now we can write that. Okay, so we can say that the image diameter, okay, the image diameter of the FOV at 40 times magnification, because from the questions at the beginning is 40x, 40 times magnification, yeah, okay. So that's why, that means that this image diameter of the FOV at 40 times magnification is actually equals to the image size, right? The image diameter of the FOV at 1000 times magnification is the same. Okay, so based on this, that will means that we will substitute this, because I is actually equals to magnification times the actual diameter. Yeah, times the actual diameter. Yeah, so that's why it is the magnification times the actual diameter equals to magnification times the actual diameter. Okay, so the magnification would be the magnification at 40 times magnification. So that means that it is at 40x magnification. It is at the 40x magnification. So at the 40x magnification, what is the actual size of the diameter of this FOV? Actual diameter of the FOV, yeah? So based on the question, this is what we find just now, right? So the diameter, the actual diameter of the FOV is actually 4 mm at 40x magnification. This is what is shown in the figure. So it's 4 mm. So that means that it is 4 mm here. That is the actual diameter of the FOV at 40 times magnification. Yeah, at 40 times magnification. So it's equals to the magnification here. This one would be those 1000 times magnification. So it's 1000 times magnification. And this actual is the thing that we want to find. This is the actual diameter of the FOV at 1000 times magnification. This is actually the thing that we want to find. Yeah, this is the point that we want to find. The actual diameter of the FOV at 1000 times magnification. So once we have this equation, then yeah, we can just solve the questions. So that will mean that 40 times 4 mm, then divide by 1000 equals to the actual diameter of the FOV. And from here, we need to convert the millimeter to become the micrometer. So to convert that millimeter, we want to convert to micrometer, we need to times 1000. So times 1000 means it will become 4000 micrometer. Yeah, 4000 micrometer. So we will do the conversion of the unit here. So that means that we will rearrange the equation. So actual diameter of the FOV is equals to 40 times 4000 micrometer. This is the one that we convert from here, 4 mm. Then just copy back, divide by 1000. So that will mean that the actual diameter of the FOV is equals to 40 times 4000 divided by 1000. So it would be 160 micrometer. So that will mean that when under 1000 times magnification, the actual diameter of the FOV is 160 micrometer, not 4 mm. Yeah, it's 160 micrometer when it is 1000 times magnification. So once we have this data, once we have this info, then next, now we can answer the questions. They want us to estimate the number of the cell that can fit across the diameter. Okay, so now that will mean that the diameter is 160 micron, right? Okay, so given that one cell, the actual size of one cell is 20 micrometer. Yeah, this is based on the calculation here. The actual size of the cell is 20 micrometer. So that will mean that across the diameter, okay, across the diameter, so it will be 160 micrometer divided by 20 micrometer. That's for one cell. So that means that it would be 8 cell in total. Yeah, you can fit in 8 cell across the diameter here. So if you want to figure out how to get this or to prevent the confusion, well, we can still do the same thing. It means that the number of cell equals to the diameter, which is 160 micron, okay, divided by 20 micron. Okay, so it will be the same. So number of cell will be equals to 8. Yeah, so this micrometer and micrometer will be cancelled out. So with that, yes, you can write in this way as well.